mics down a little bit because some slides will be anatomy and you need that.
Okay. Well, good afternoon. Yeah, thank you very much, Ian, for the kind invitation. And I'm really very glad to be here. Also, thanks for this named lecture. I'm very glad because really this is a renowned Canadian Center for the Behavioral Neuroscience, home of numerous quite distinguished investigators with really a record of very influential work on animal, especially rodent behavior. And as we all know, neuroscience is a highly interdisciplinary field ranging from molecular to developmental to cognitive. But many young aspiring neuroscientists here, I suppose, enter this field with initial fascination on brain mechanisms of behavior. But the complexity of the problem usually bring us to highly specialized subdisciplinaries to take reductionist approaches, especially filled with technical advances in molecular biology, microscopy, optogenetics, that give us increasingly higher spatial temporal resolution of the brain parts. On the other hand, to understand the behavior, we actually do need to study behavior itself, as people here know all along. So as we increase our resolution of cells and circuits of the brain parts, it's increasingly appreciated that we also need to have increasingly higher resolution of the behavior analysis, the sort of movement motifs sequencing ethogram, if you will. So I actually sort of, in a way, agree with this view in this article. I think many of you might have learned that, careful behavior analysis hopefully will allow us to have some insights of the computation of the algorithm of the system. And that's when this neural implementation can be studied and the most informative. So we took a detour to cellular developmental studies, but recently we begin to explore the circuitry mechanism of motor control. So currently, the overarching theme, if you will, is to try to understand the principles of the organization and the assembly of neural circuits in the cerebral cortex in the functional context of motor control. So today I'll cover a range of topics. First, a relatively brief summary of building genetic tools to target neural circuit elements from different subpopulations to the so-called cardinal neuron types. And a brief highlight of our recent findings that one of the best description of a neuronal cell type, we believe, may be their synaptic communication styles. And this may be rooted in the transcriptional architecture of these cells. And also recently, we identified a chandelier cell pyramidal neuron microcircuit module in the cortex. So I'll describe our progress in understanding the specification, the sort of subtype granularity, and the connectivity of this interesting module. But I do want to spend quite some time of our recent progress in understanding pyramidal neuron, glutamatergic pyramidal neuron types in the context of cortical control of skilled movement. So just a few words briefly. The cerebral cortex consists of really a constellation of functional areas that form a representation map of the external and internal world. And these areas are strategically connected to integrate multisensory information with internal goals and memory, and to make decisions and to configure plans for action through the multiple subcortical channels. So classic studies using imaging and others have delineated a macroscopic connectivity pattern of different brain areas. But we know ultimately the brain function emerges from dynamic neural circuits from millions of neurons communicating at synapses at millisecond time scale. So really the enduring challenge is to bridge across these different levels of organization. And individual cells communicate among different areas at synapses. So these inherently provide potential bridge. These are the basic elements of neural circuits and networks. They are also the basic building blocks of developmental assembly. Importantly, they are the unit of gene regulation, where genomic information can unfold to organize and assemble circuits. So, of course, the challenge is the vast number and the diversity of neuron types. And this has been a formidable challenge to understand the cellular architecture of cortical organization at higher level. So despite the functional and cellular complexity, the basic organization and construction plan of the neocortex has evolved over 100 million years ago, and this plan has been conserved across mammalian species, even in light of the more recent elaboration in primates and humans. And what do I mean by this basic plan? So this is, in my mind, analogous to the basic body plan, which can be traced to early invertebrates, and it is rooted in these Hox gene clusters along the chromosome. So the basic cortex plan can be more apparent with also a species comparison. So in the mouse, there are recognized about 40 cortical areas. The location of these areas and their connectivity is largely conserved to those of human. And obviously, the laminar organization of cells not shown here. And then the characteristic triple descending pathway featuring the cortical striatothalamic loop is well conserved, including some of these local circuit templates and motifs. And these are implemented by a large set of what we call cardinal neuron types, such as the cortical fugal pyramidal neurons, the axonic chandelier cells that I'll elaborate. And intuitively, this basic organization plan of network scaffold or circuit templates are nearly identical among individuals of the given species. So they need to be reliably generated through developmental programs. And ultimately, this program has to be encoded in the genome and unfold reliably and selected through evolution. So these days, the idea of deep learning and the artificial intelligence is in vogue. So in a way, perhaps we can think of the genetic program of this network scaffold and circuit template or cardinal types as the product of a very deep evolutionary learning by the species to deal with the invariant features of the physical world for perception, or the musculoskeleton structure of the species to generate movement, or even some of the conspecific social relationships to manage social interactions. So in this context, one of the at the core of understanding this brain organization is really the issue of cell type. What are the large array of genetic cardinal types, and how these are then customized through development or activity or experience to circuit elements that solve behavioral problems. So in the cerebral cortex, despite the large number and diversity, there are two broad transmitter classes, the glutamatergic pyramidal neurons So with large dendrites and axons, so these form the skeletons of the local circuits, and they form all the inter-areal processing pathways and all the output channels. But their diversity is vastly underestimated because in most cases their axons are cut. We don't know where they project to, who they talk to. So that's a major deficiency. And the other 20% or so GABAergic interneurons, despite their small number, has more apparent morphological diversity because you can see their dendrites and axons. So they play important roles in balancing excitation, gain control, network oscillation, so on and so forth, and different subclasses specialize to perform these different circuit operations. So in order to recognize systematically these different neuronal classes and to understand their connectivity and function, we need really a genetic toolkit with two major important properties. One is specificity at an appropriate granularity. Is there a question?
Is this in mouse? Or
Pardon?
is this rat? Is this in mouse, rat?
It's either. So both. This will be
So
rodents, but primates. It should be in all these generic types, so it will be, conserved. Although there are larger number and probably more diversity in primates, but the cell types. Hmm?
This is just generally speaking.
Yes, although most of the knowledge, more are now recently from rodents, but I think, the general idea and the concept is. But in terms of genetic toolkits, yes, it is in mouse. We need, one is specificity, one is comprehensiveness. And I think a specific targeting of a neuron type is sort of analogous to cloning a gene in genetics. It will provide the clarity of an answer that is not achievable otherwise. Often, dealing with a mixture of cell types generate confusion. And the other is if we can target and manipulate multiple or odd cell types, that give us the opportunity to systematically understand their connectivity and operation. So very briefly, this is a summary of our over 10-year plus effort. So recognizing the significance of this problem, we begin to use mouse genetic approaches to target subpopulation of cortical neurons. And this is based not only on the molecular markers that we can identify are available, but also the developmental genetic logic. So all the cortical neurons are generated from progenitors lining the embryonic ventricle wall that contain different groups of progenitors. These are distributed according to transcription factor expression. So in the dorsal pallium, these generate all the glutamatergic pyramidal neurons. In the so-called lateral ganglionic eminence, they generate all the GABAergic projection neurons of the basal ganglia. And then the medial ganglionic eminence generate the interneurons, GABAergic interneurons of the basal ganglia and the cerebral cortex. So we use either molecular markers or transcription factors and their combination to design strategies to target these cells and the lineage. So in the first round, we generated multiple mouse lines that target major populations of the GABAergic neurons. But we recognize that there's no simple correlation between one gene and one cell type defined by their morphology, physiology, and so forth. So in the second round, we use various combinatorial approaches to combine 2 or more genes, not more than 3, and to use viral vectors combined with driver lines to engage their anatomy, but also to engage their developmental program, meaning their lineage and their birth order. So this turned out to be a very important strategy to target more specific cardinal types, such as these so-called chandelier cells. And we have made progress now along this using more systematic fate mapping in the MGE and also LGE. And more recently, with the advances in the molecular biology of the glutamatergic pyramidal neurons, in collaboration with Paula Allada at Harvard, we have made progress in targeting various types of glutamatergic projection neurons and also their lineage. So in this recent summary, I suggest that the specificity of the cell type access, the technical issues are largely actually solved. And this comprehensiveness of accessing multiple cell types need a larger scale effort that we now fortunately are able to join the US Brain Initiative cell census network that continue to fund this research. So I've been talking about cell type in the sort of more technical aspect of experimental access. But what is a neuron type and how should it be defined is actually a very contentious issue. In fact, there's debate whether there is a biological basis or it's always going to be an arbitrary grouping. So, and in this recent, again, opinion article, the difficulty of just registering anatomical features, molecular features to functional features is the major challenge. And I think many people here recognize the work of Bruce McNaughton and others, the hippocampal place cells, grid cells, and others. How do those correlate to functional types, correlate to molecular types? And I would argue that many of the important issues that debate in neuroscience can be traced back to the ambiguity of these cell types under study. And so this is not just the issue in neuroscience, even in genetics, the idea of what is really a gene is continually to be discussed. So although in smaller animals with smaller neurons and more stereotyped morphology, there is sort of a practical consensus on what is a cell type. In the larger brain, this sort of seemingly intractable variability or diversity brings up the discussion whether cell type is a real biological or it's always arbitrary and operational, and whether a continuous variation of phenotypes or distinct groups are the better description of these entities. And also, are they conserved across species? And so in most cases, this diversity is a sort of in-your-face problem. Anytime when you want to study a circuit, you want to know how many cells are there. But we think even more fundamental issue, or equally fundamental, is what is the identity of a cell? How do you define a cell type? And this is the basis for understanding diversity and ultimately their connectivity. And this is a difficult problem because individual neurons are, in a very real sense, sort of individual microorganisms in the cellular society. So they have multiple different features and vary in multiple dimensions, classically in the shape, but certainly in their physiological properties, in their molecular expression, and they all have a developmental history, and they mature and adapt, and ultimately they are only useful if there is a behavioral function that they can perform. So this is sort of a classic analogy that neuroscientists have been blindfolded for many years, and each is looking at a different piece. But more recently, and technical advances allow us to look at multiple features, but also in a quantitative and higher supered way. So certainly to look at gene expression in individual cells, but in many of them, and to quantify these expression patterns. And also in understanding their morphology. I'll show you one example on chandelier cells in quantitative and systematic way, in understanding the developmental basis of these and ultimately function. So I'll quickly highlight this study from high-resolution single-cell transcriptome analysis. So in this case, we, using combinatorial genetic strategy, identified or purified in a way 6 different types of GABAergic neurons in the mouse cortex. So these include the so-called axonic chandelier cells that target the axon issue segment of the pyramidal neuron. The parvalbumin basket cells target the parasomatic region, and the dendrite targeting cells, the interneuron selective target cells, and so forth. So the reason we spend a lot of effort is that there has been over 3 decades of study on their anatomy, their physiology, and their developmental study. So this gives a very compelling context to interpret the molecular expression data. And then I won't go through this technical aspect, just to mention that we developed a modified method that allows us to give very high resolution transcription profile in these individual cells belong to these 6 populations. And then using a computational screening strategy, we systematically compared the gene expression of these 6 populations and specifically asked the question, what are the gene family expression profiles that distinguish these cardinal types? So you may know that in the human genome, the genes are classified into about 600 or so gene families, from metabolic genes to ion channels to transcription factors and so on and so forth. So we found with those 6 cardinal types, there's only about 40 gene families that very well distinguish these cell types. And these 40 gene families really boils down to 6 types, 6 kinds. This is the cell adhesion molecules. In fact, different subfamily of cell adhesion molecules are highly differentially expressed. The transmitter receptors, modulatory receptors, ion channels, signaling molecules, and here importantly, the neuropeptides. So in addition to the well-known somatostatin, NPY, and so on and so forth, these populations in addition express 6 or even a dozen different peptides. So in a way, they speak different languages or different accents, and then there are release machineries that release these peptides because they are different from transmitters versus small synaptic vesicle versus dense core vesicles and all that. And then finally, there are transcription factors. So these genes are all, except the transcription factors, form a molecular scaffold along the cell membrane. And we think of them really as 3 kinds. One is related to connectivity, which are cell adhesion molecules. And the others are the input, which is the receptors, the signaling, the ion channels, and then the output, in addition to the substance they release, what they speak. So ultimately, we use the word synaptic communication. So not only who you connect to, but how do you talk to or how do you listen? And we think that this property, cellular property, is key for defining that cell type. And interestingly, there seemed to be a genetic recipe in those cells that systematically tune those features using a relatively small set of gene families. And for example, to build a fast spiking interneuron, the cell will use fast receptors, glutamate receptors, GABA receptor for fast input. They will use fast channels for fast spiking. They will use fast release machineries for output. And this sort of a congruent selection of these molecular components most likely is generated through transcriptional regulation. And we think that this overall framework of synaptic communication includes the more traditional description. For example, morphology really is a reflection or a proxy of connectivity, trying to reach a particular input with a particular shape of dendrites or try to communicate to the targets with axon. And the physiological property really ultimately is to mediate input-output transmission, again, using receptors and release machinery and all that. So one of the issues that we didn't touch upon here is that for these 6 populations, there are further diversity within, for example, basket cells and others. And they look different, and people wonder. So there's this issue of continuous versus discrete variation. What is the appropriate granularity? And here I show this diagram, a comparison between retina and the cortical neuron. So in the retina, there are several levels. The photoreceptors, the amacrine cells, the bipolar, the retinal ganglion cells, and they are hierarchical. So even for this bipolar cells, it is now recognized that there are probably 15 types, and that's probably saturating. And this is based on morphology, connectivity, transcription profile, even function. And the problem with the cortex is that in any class, when you look into, you see further variations that seems sort of intractable. And so this issue of, at that point, is that there are just some random variation, or there are further sort of logic of discrete subtypes. And so that's, again, an important issue. What is cell identity? What is subtype? So here I use this example of chandelier cell to address this issue in the cortex, because chandelier cell is one of the most well accepted and better described cardinal type. So for those of you who have not heard, this is a type that specifically control the axon 2 segment of the pyramidal neuron only. And this is the region, of course, action potential is generated. So one chandelier cell will target hundreds of pyramidal neurons. And because of this, Francis Crick actually coined the term veto cells. Like, this cell may have the potential to silence hundreds of pyramidal neurons regardless of the input. So in a way, unfortunately, Donald Trump might have that power. So a number of years ago, we, using genetic fate mapping, discovered the developmental origin of these chandelier cells. And here is an example showing this red chandelier cell, and the green is the molecular marker of the axon 2 segment of pyramidal neurons. The cell body are not shown, but you can see that in every case, it targets this very specific location. But even early on, we found that there are chandelier cells in different layers, layer 2, 1 border, layer 5, layer 6, with different dendrite and axon. So are these just some haphazard variation, or there are different distinct groups? So to address this question, our approach is a higher resolution complete morphological reconstruction and analysis. So in this case, it's not easy to do, again, in larger brains. You need to solve several challenges. One is to label individual cells very reliably and to completion. So we mostly solved this problem by combining genetic targeting with viral targeting. And then you need to image these cells at axon resolution of the whole cell, and in projection neuron, that means usually the whole brain. So this has been solved, or are being solved by the Genelia mouse light project using 2-photon microscopy, or by my collaborator Qingming Luo's lab in China using the so-called micro-optic sectioning tomography. I'll show you an example. And then we need to reconstruct these cells in completion, and we need to analyze them in register them in the framework of connectivity. So here is an example where during this imaging procedure, the brain is embedded in very hard resin, and then using a wide field CCD camera to image in a stripe fashion. And then once that surface is imaged, it's cut in 1 micron section and then imaged the next. But along the way, this proprotein iodine stain every cell body position, and then these are the genetically labeled chandelier cells. So this give us the full morphology of individual cells, but the blue dots give the location of each cell, so you can register precisely the labeled cell in the background of the mouse brain. And here is an example showing this imaging, and this is actually unprocessed. You can see these stripes are the imaging plane. It's like taking a picture from the airplane. And then you can see this is the layer 1 to border the chandelier cell, and then these are the dendrites, and these are the axons, and then you can see individual boutons registered in the cell body position. So
What layer is the cell body in?
the cell body in here is at the layer 1-2 border
Layer 1, 2.
in the prelimbic or prefrontal cortex, but there are other areas.
What was the scale bar? Just out of curiosity.
Scale bar. The cell body is about 15 micron, and the bouton would be about 1 micron or so.
You're
Worry
studying
about
those things, David, you don't even know it.
I'm wondering about the extent of the whole.
Oh, the whole, yeah, so that would be about 300, 500 or so with some variation. So yeah, you can see these are all individual cells. This will be about, half micron or so, yeah. So you can see that this is in the prelimbic motor and the somatosensory cortex, and there are, they look different, but not only they look, so there are layer 2, 1 border, layer 5, layer 6. And what's interesting is that, look at here, the axon, even for the layer 2, there are axons in the layer 2 only, layer 2, 3, and layer 2, 3 to layer 5. So because these axons really targeting the pyramidal neuron axon issue segment, we think this actually is functionally relevant, that they are actually controlling different subsets of pyramidal neurons. And then you see that in these layer 5 cells, typically their dendrites are located within the same layer, but there are ones that goes to layer 1. Again, they actually grab the pia, so these ones really want to get inputs from layer 1. So you can infer that these cells are not just in different locations, they they have different input-output. And we think that they are different, these suggests there are distinct subtypes of chandelier cells that with the different input-output connectivity, for example. And this reminds us again of the retina, where different bipolar cells with different sublamina actually indicate connectivity. So we also have evidence that these different upper, lower chandelier cells have different molecular markers that I want time to show further, that we actually have done now a lineage fate mapping and show that there are 2 possibilities. One is that when these cells are born, they are all the same, and then they migrate, and depends on when they end up, they take up these different features due to local cell-cell interactions. The other is, even in the very beginning, there's already genetic information, and our evidence actually favor the latter, that even in the early development, they also already begin to have that information, suggesting, again, there are likely distinct subtypes. So finally, these layer 2 chandelier cells, they don't just connect to any pyramidal neurons. They actually connect to pyramidal neurons based on the projection pattern of ensembles. So in the prelimbic cortex, in the layer 2, 3, there are at least 2 types of pyramidal neurons. The green one is projecting to the basolateral amygdala, the blue one projecting to the contralateral cortex. And again, using retrograde labeling with CTP, you can label these 2 pyramidal neuron population. And then using genetic labeling, we can label this population of chandelier cells that are especially late born at E17. And Jianteng Luo and the PhD student Tusi Rong show that there's this very surprising selectivity where this chandelier cell selectively innervate the BLA projecting pyramidal neurons, but not the contralateral projection neurons, both in terms of probability and the strength. But in terms of input, it's reversed, where they receive input from the contralateral projecting neurons, but not from the basal BLA projecting neurons. And further, these are just rabies tracing and optogenetic data, but the summary is that this local connectivity motif is embedded in a larger brain network because the BLA projecting pyramidal neurons receive reciprocal input from BLA neurons, and there's a reciprocal loop here. And then the contralateral projecting pyramidal neurons is in another reciprocal loop of contralateral pyramidal neurons. And in terms of the long-range input to these chandelier cells, again, they receive input from this loop or this network, but not from this. So it's sitting in a really key position to regulate both the local ensemble, but also the more global network communication. And so currently we are using better tools now to try to understand the function of these cells in a more in vivo behavioral setting. But this just give my summary that this is our approach, is to be able to track these increasingly identified cardinal types or even subtypes from their developmental origin to their connectivity. And I think the key, again, is are these really meaningful when we try to divide them to this kind of granularity? Do they matter in any kind of behavior? And in that case, we really do require the similar or matching level of granularity of looking at behavior, looking at the ethograms. So with that, again, briefly, even to understand chandelier cells, we need to understand the pyramidal neuron ensembles that they control. And really, it's the pyramidal neurons that receive information, process information. So that's why 5 years ago, we invested significant effort to genetically target different population of pyramidal neurons based on laminar gene expression. Again, the lineage here is just a few examples to show that we are able to recognize these layer 6 corticothalamic neurons that has their dendrite here, which project to thalamus, and the layer 5 corticofugal neurons, which are these blue lines here that mediate all the output to spinal cord, superior colliculus, pons, and so on and so forth. And then the corticostriatal neurons in layer 5A, and cortical neurons in upper layers, so on and so forth. Then by using viral injection, we can begin to restrict analysis to specific cortical areas and begin to see their projection targets. And we can use these two-photon tomography to look at viral injection here and then all these projection patterns at the population level. But ultimately, we need to actually also carry these analysis to single cell level. So many of these may still contain multiple components and channels. So this is very much ongoing. So to this audience, I really want to present in the last 15 minutes or so our recent progress in understanding or really exploring cortical control of movement. And I think many people might have seen this from Daniel Walbert, that there are many fascinating brain functions, but the real reason for the brain is to generate adaptive and complex movement. And among those, really this skilled falling movement, and this should be very familiar here, really evolved from early vertebrates and elevated in rodents, but really occupy a singular position in primates and humans, where there are all these fascinating movements that I really don't need to elaborate here. So this forelimb is a complex organ with 20 or so bones and a similar number of muscles and tendons, and generate very complex movement, many degrees of freedom. So although there are progress in building robotic arms, the sort of dexterity is far from the biological arms. And really the question is how does the brain control dexterous movement, presumably by precisely activating many different muscles at precise spatial temporal patterns when involved in different tasks. So the rodent, I think, again, this audience really is not needed for this, but has been underappreciated for quite some time. But I think probably to a significant extent of Ian's pioneer and persistent work, I think it's going to be it's really going to be a revolution here. So I buy the idea that it is probably involved relates to the to eating and to food handling, and there's similarity of when you train them for reach and grasp with human, and including their musculoskeleton and including the way they use digit. And although all the motor neurons are in the spinal cord that control these muscles directly, the question is what are the role of the brainstem and also the cerebral cortex, presumably in higher level coordination. So this role of motor cortex in motor control really is not well understood. And in fact, there has not been a very compelling framework. Although there's evidence that the motor cortex is involved, and there's recordings dating back to Edward Evers, and also this very intriguing but controversial work that long duration stimulation can generate rather complex movement. It's only quite recent that Adam Handman, using more modern tools, show that the cortical activity is both necessary and sufficient for initiation and execution of the movement. However, the underlying circuitry and cellular basis really is unexplored. In fact, there's rather different ideas of what the motor cortex might be. So this is very different from the sensory system, where in the visual cortex, dating back from Hugo Wiesel, that we know there are labeled lines from the receptors, and there are specific types of cortical neurons for orientation, direction, and the speed, so on and so forth. But once in the motor cortex, it's not clear what features of the movement, whether it's direction, kinematics, force, so on and so forth, are important, and how these are represented or coded by neurons. So it's even debated whether there are specific regions or types, not to mention circuits, or there's this rather more popular view from mostly primate studies, where there are broad population dynamics, statistic interactions along a large swath of the system that actually, in some very more abstractive way, ultimately generate movement. So we start with no sort of pre-assumption, but I think an important point here I'll emphasize is a mentality and the approach of screening. And our approach is to, in a way, to do a sort of a small-scale genetic screen of cell types, because we generated dozen or so mouse lines that target these different pyramidal neuron types. So we can, without assumption, look at one by one. And the other is a optogenetic screen of areas. So again, there is the idea of motor areas, but based on rather classic stimulation or lesion studies that has a lot of caveat. So we use this optogenetic stimulation in head-fixed but scene-skulled awake mice. So in a grid pattern covering much of the frontal cortex, including sensory, motor, and beyond. And then we look at behaviors, initially the forelimb behavior, but more recently the orofacial behavior. So the idea is to see what happens. And what happens has been quite fascinating. So the first is this FESF2 corticofugal neuron schematic is drawn here, that it goes all the way down to spinal cord, pons, cervical, many areas. So by stimulating this population in this grid fashion, so each grid is about 300 micron into 3 by 6 millimeter area, we showed not only that you can generate prominent movement, but that there are, in most cases, 4 major trajectories, and these are organized along this medial posterior to lateral anterior axis. So here I'll show you a few examples. So in this blue dot here, you see this mostly lateral backward movement with an open arm. And then in this green area, you see this repetitive movement, again with an open arm. And then in the light blue cyan area, you see this upward forward movement, but now you see a partial closing of the hand, resembling perhaps a reach type. And then in the red spot, you see this most complex, where the arm is lifted, but with a turning of the wrist and the closure of the digit, and then the orofacial movement. And these are the movement trajectories of different cases. So if we average from 13 animals, these are the projection of the trajectory. You can see that although there are potentially many different freedom, there are mainly these 4 dominant trajectories that occupy a relatively restricted space. And interestingly, this trajectory is topographically organized considering the corticofugal pyramidal neurons along the cortical surface. So again, these trajectories are generated here, these are generated here. And so there are different ways of plotting this to look at their movement. Here is another way to just measure the endpoint distance to the mouse, and you can see the shortest is here, the long is there. And this trajectory is also correlated with the complexity of the movement, whether it's only the forelimb or forelimb and the hands and then mouse, so on and so forth. The other thing that's interesting is that now when we look at the corticothalamic population, which ends in the thalamus, we also generate various movement and with a similar map. And for the interest of time, I won't go through of these. But compared to the FESF2 map, it's shifted to the more lateral location. And overall, the movement is more jittering. And the hypothesis not being tested yet is that these may activate thalamic neurons that project back to the cortex that might engage these FESF2 neurons. And perhaps this may be involved in the continuation or sustained movement through sensory motor feedback, so on and so forth. The most striking movement we induce is this plexin D1 corticostriatal and corticocortical population diagrammed here. So we actually didn't see any movement in the head fixed condition, at least in these other areas, except this one red spot. And you can see here, it is a highly complex movement involving both arms, the lifting, the full supination and the closure of the digits, and then the rhythmic orofacial movement. So this is an area that actually new. It's not the sort of rostral or caudal forelimb area or the ALM area recently described by Carl Swoboda and others. It's in this red spot here. And so if we overlay these 3 cell population, this is sort of what it looks like. The green is the forelimb movement. The red is the orofacial movement. And you can see there's the forelimb only area, the orofacial only area, and then the both coordinate area in FESF2, similar in TLE4, but in the plexin D1, only this sort of this coordinated area. And so if we want to consider cortical map, it's really a map that is differentially implemented in different layer, but more importantly, probably different cell types. So I'll spend a little more time in the last few minutes on this hand-mouse synergy in the context of eating. Oh, okay, so one more example to show this the plexin D1 mouse. In this case, we An Xu in the lab first mapped this particular area, because it may vary a little bit in different mouse, in the head fixed condition, and then he put implant optic fiber to look at the effect in free moving animal. So you can see, let's see, to the left. Yeah, so this animal is rising, but then the light goes on, and it stops the movement and then adopt this feeding-like behavior. But what's interesting is that in the head fix, you only see the uplift of the arm. In free moving, you see the lowering of the head and the posture. So it looks like this whole eating posture is inserted in otherwise another behavior sequence. And then here's another interesting example where the animal is going to scratch the head with the hind limb, and then the light turns on, and then they adopt this behavior. And when it's off, it goes back. So to our knowledge, this is one of the really striking case. There seems to be a command function, this layer 5A population, probably a couple hundred cells, and maybe we can even do with less. So very intriguing. And this is resembled eating, and I sent this video to Ian. He was intrigued. So eating, starting with using the mouth without even jaws, it's filtering. And then using the mouth, you can bite and chew. And this goes even to the carnivores. But really in rodents and the primates and us, we engage hands. And there's this more sophisticated sensory motor coordination in people call hand-mouse synergy. So again, this I think everybody knows. And we are interestingly, can recapitulate this behavior in a head fixed, where the animal now pick up the food pellet with the tongue, but then invariably transfer to hands and to manipulate and eat. So this gives us an opportunity to look at the circuitry mechanisms. And we show the stimulation, and one of the key questions is whether this area that we call lateral motor cortex, or MC, is actually needed. So the evidence is yes. You can look at more closely that they pick up the food pellet with tongue, and then they never really just bite and swallow. I think Ian said this has to do with the size of the pellet. And then they use both hands to bite and turn and then bite, this sort of alternate bout until they finish. And sometimes you can see maybe even a leading hand or a following hand. So when we use Musimo to inactivate this area, you can see that they can still pick up, but in the contralateral side, this hand now is handicapped. It can lift, but it doesn't turn and close to help with the other hand. And in other cases, you can see that it managed to use the other hand. So this, again, is the contralateral hand. And it managed to take one bite. And typically, it'll, turn and then bite again, turn and bite again. But you see, it seemed to freeze here, and not that it's moving. We think it's really trying to come on. It's worth the waiting because then it fails. So now we are doing cell type resolution, cell ablation, and others of the plexin and the FSF2 cells. and this is ongoing. And also, obviously, the goal is to record and then to image these cells and others. So with these tools, it allows us to really get to the neural circuits. So we can use viral injection in this area to look at their exon projections. I already said that the plexin D1 cell project to ventrolateral striatum and several cortical areas that I'll elaborate. And FASF2 cells project to all the sort of subcortical regions from the spinal cord to the pons to the supracolliculus striatum thalamus. And TLE4 only project to the thalamus. So using rabies tracing, we can look at their input. Again, the fascinating thing is that all these three populations in the cortex, they project to only several areas, sensory motor, primary sensory motor areas, but they are all related to the forelimb and the orofacial region. And for the plexin D1, it goes further to the insular cortex. And I think I'll just show you the summary here. So the color are three different cell populations, and this is the output. So these are the small number of areas. And then in terms of the input, it's reciprocal. So most of these receive input from them again. And then the receive input from the thalamus, which is a major area, the raphe, and then the pallidum, surprisingly. And then the output, the plexin D1 is mostly to the ventrolateral striatum, and then the FASF2 is to all the subcortical area. But what's interesting is this real emphasis of the orofacial and the forelimb area that is implemented really at the level of hardware. So the last slide, I think many people know this very well. We are very excited that this work that Ian really pioneered probably 30 years ago now is increasingly used in head fixed and free moving from multiple different behaviors, but with the increasing sophistication of the cell type tools that we can observe them, we can record, we can perturb, and we can do it in a wide field imaging to look at new areas, and we can do it at cellular resolution. And also that many of these interesting behaviors seem to be conserved not only in the mouse but also primates and even in human, where this presentient gyrus can induce, again, hand-mouse synergy. And ultimately, I think many people are interested in relevance to disorders. So this movement really is a reflection of the mental state, and the deficiency also is probably a sensitive indicator. So these are the people who did the work. Anirban Paul is a postdoc that spearheaded the single-cell transcription analysis of the GABAergic neurons. Jiangteng Lu and Jason Tusiarong are postdoc and MD PhD student studied the chandelier cell connectivity. An Xu and Hemas Mohan are two brave system neuroscience postdocs that has been studying the mouse motor cortex. And thank you for your attention.